right, now we're going to cover chapter 13. We're going to focus in on viruses, viroids, and purons here. When you're looking here at the viruses, viruses are considered acellular, non-living particles. So we talked about back in chapter 4, prokaryotic cells, which are cells without a nucleus and organelles. In chapter 12, we hit on eukaryotic cells, which have a nucleus and organelles. And now we're going to go to something that's considered acellular, no cell. Okay, because of this, these guys are obligate intracellular parasites, meaning they have to gain access into a cell in order to reproduce. All right, so they have to hijack or commandeer the cell in order to make more viruses. Now, this is an issue because they're not alive, but they do need an alive or active host in order to be activated, and they can cause infection. The word virus is Latin for poison. So let's look at some basic viral structures that are here. When we look at a virus is always going to have an internal nucleic acid core. This nucleic acid core could either be DNA or RNA. Another thing to note that is it could be a double-stranded DNA, it could be double-stranded RNA, it could be single-stranded DNA or single-stranded RNA. It has lots of variations of either having that DNA or RNA molecule. Now, one thing to note though too is that it only has one type. It does not have DNA and RNA, okay? It has one or the other. Now, this inner core is going to be protected around with a outer protein capsid. These capsids can have different kind of shapes to them. They can be very complex. They could be spherical. They could be a rod. There's lots of different ways that they can have this capsid. And then some of these viruses also have an envelope. The envelope is optional, but an envelope does offer protection for the virus. Just like an envelope does for your letters, it makes it where it's hard for you to see what's inside of it. This envelope offers protection for the virus to hide from your immune system. So let's do a quick comparison of viruses versus bacteria. So when we look here, a bacteria has a cellular organization because it does have cells. Viruses do not. Bacteria also do metabolism. They are able to create ATP and do chemical reactions. Again, viruses cannot. Bacteria can respond to its environment. Viruses cannot. And so when we look at here, this is what makes bacteria living versus viruses non-living. Another thing though is that they can replicate. Bacteria replicates through binary fission, whereas viruses are going to replicate only within a host cell. Whether that host is a bacteria, plant, animal, a protist, fungus, but it has to gain access into a cell. Bacteria can also mutate. Because it mutates, it can make changes, which makes it hard for us to potentially treat with antibiotics and things like that. Viruses also have the ability to mutate. This makes it difficult to develop effective vaccines against viruses and be able to protect against them. When we're looking at bacteria, because it is made up of a cell, it is going to have a plasma membrane. It is going to have ribosomes. It doesn't have the other organelles, but it does have ribosomes. It has DNA and RNA. When we look at the virus does not have a plasma membrane. It does not have organelles at all. And it's either going to have DNA or RNA, not both. When we look at bacteria, it talks about if they're filterable, size dependent. No, they're not when we look at this. But viruses are, because they are so small. They're way smaller than bacteria are. And then bacteria are sensitive to antibiotics. However, viruses are not. Viruses are going to be more sensitive to interferons. All right, so there's different chemicals that bacteria are going to be sensitive to versus viruses. So let's look at the general morphology of a virus. There are different types of viruses. There can be what we call helical viruses. When you're looking at helical viruses are long rods. They can either be rigid or flexible. All right, so some examples of some helical viruses are things like rabies, Ebola, hemorrhagic fever. So these are just some examples of some viruses that are helical. We also see that some are what we call polyhedral viruses. Now, polyhedral viruses, they're going to have like 20 sides to them. All right, so they look like a sphere, but they actually are a combination of lots of like small triangles kind of put together. So some examples of these are adenovirus. Adenovirus is the virus that causes a common cold, and then poliovirus. Some viruses are enveloped. If they are helical enveloped viruses, this is going to have that helical structure with an envelope around it. That's what you can see here with D. This is going to be like influenza, which causes the flu. We also have some envelope polyhedral examples here. This is going to be like herpes simplex. So it has a polyhedral structure, but it's inside of an envelope. There's also some other complex viruses. These complex viruses include pox virus and bacteriophages, and this just means these viruses are more complex with their structure. Bacteriophages are really unique because bacteriophages are the ones who infect bacteria. So if you look here at this picture, you have the pox virus, and then over here, this guy is the bacteriophage. If you've ever seen Toy Story, the first one, when Woody gets stuck in Sid's room and all the toys come out that have been like mashed together, this reminds me of the one that crawls out from under the bed with all the legs and the doll's head. That's what this one looks like. It's a pretty scary type of virus. However, this one is going to infect bacteria, not us. Now, with viruses, we do not use the normal taxonomy that we use for bacteria and plants and animals because these guys are considered non-living, but they are grouped by their type of nucleic acid, whether they're a DNA virus or an RNA virus. They're also going to be classified by their morphology. What do they look like? Are they helical? Are they polyhedral? Are they enveloped? That's the next one, presence or absence of an envelope. And then also their host range. Who can they infect? What are the types of cells that they can infect? This is their niche. Most viruses are very specific on who they will infect. So when we look at this, we have the genre. This is going to be the name suffix of the virus. And so example here would be norovirus, denotes a viral genre. We also see we have the family name suffix, which is viridae. So you have the Caliciviridae denotes a viral family. So we can see that we can use some terms to help put these into groups. There is a table, 13.2 in your textbook, that talks about these host niches or ranges of viruses. So since viruses are non-living, it makes it a lot harder for us to study them in a lab. All right, and so because of this, if we want to culture them and cultivate them to grow, it is going to take a few more steps to do this. So cultivation of bacteriophages, remember bacteriophages are the ones who actually infect bacteria. We can use a plaque method for this. Okay, with the plaque method, you use a nutrient auger. You then take a bacterial, do a bacterial spread plate. A spread plate is where you take it and you culture bacteria around the whole surface, so the whole surface is covered in bacteria. It allows the bacteria to grow. Then you're going to add a thin layer of liquefied agar. This liquefied agar is going to contain the bacteriophage. It's going to contain the virus. Okay, then we're going to incubate it. It allows the bacteriophage to infect the bacteria. It then can start to replicate and kill off the bacteria, and this creates a clearing. These clearings are known as plaques. This is where we would then count the plaque forming units. So you could count how many there are. Each of these areas here where there is a clearing, it would be representing one bacteriophage. Now this method is pretty easy because it does allow us to be able to grow bacteria quickly and get results quickly. But if the virus does not infect a bacteria, this creates a little bit harder opportunity to cultivate it. So there are some animal virus cultivations that we can use. We can put them into living animals. When you put it into living animals, of course, it's going to end up potentially making the animal sick, but it allows us to look at the signs and symptoms that go along with it. We also can inject them embryonated eggs, meaning that the eggs have started that development process. So there's several layers of tissue. This will let us know where the viruses prefer to replicate, what types of cells. We also see that we can do some cell cultures. When we do the cell cultures, we're going to look for what we call cytopathic effects, or CPEs. This is going to be where we're going to see where those cells have been affected. Cell deterioration. Now, one thing we can do is we can do a serological ID of viruses, and these can be very specific and let us be able to tell what virus is present. If you've had the flu or you've been tested for the flu, this is like what they do. So they take the swab, they place it in your nose and collect a sample. On the swab, if the virus is present, there will be virus antigens there. They then add several drops of a liquid. This liquid contains antibodies. The antibodies, if they come in contact with the antigen, are going to react, and it normally causes a color change. This is why they can then tell you pretty quickly in the clinic if you have the flu, and even if you are positive for type A or type B, because these antibodies are going to be very specific to that specific virus. Another thing we want to look at is the viral multiplication process. How do we get more viruses? Now remember, these viruses have to invade a host cell. When they invade the host cell, they commandeer the host metabolic activity. So what they do is it's almost like they hijack the cell. They tell the cell, you're no longer going to perform your normal functions, and you are now going to make more of me, which is is the virus. All right, so they're going to make more of this. Another movie reference is if you've seen the Pirates of the Caribbean, Jack Sparrow is always commandeering or taking over ships. He then uses the ship for his purpose. This is what the virus is doing. It's taking over the cell and it's causing the cell to make more viruses. A single virion is capable of producing thousands more viruses. All right, so it can end up multiplying very, very quickly. Now, there are two main cycles or ways that the virus can accomplish this goal. The first is called the lytic cycle. Now, the lytic cycle has 5 main steps to it. So let's take a look at these really quick. And this is found in your textbook. So if we take a look here, the virus first has to attach to the cell. This is the attachment phase. This is where, in this case, the bacteriophage attaches to the cell. Once it attaches to the cell, it now has to get its DNA or RNA, its nucleic acid, into the cell. This is called penetration. So it's going to do penetration, allowing its nucleic acid into the cell. Once inside of the cell, it's going to start what we call biosynthesis. This is where it's going to hijack the machinery of the cell. It's going to use the ribosomes and those structures to make more pieces of the virus. Okay, so it's going to synthesize the viral components, the capsid, more DNA or RNA, and all of those structures. Once all the structures are formed, it goes to step 4, which is the maturation. This is where we're going to put all the pieces together. The viruses now are going to be mature and ready to be released. Okay, this is the last step. The last step is where they release all of the viruses. Now these viruses can go and infect other cells. All right, and so in this case, a lot of times when we look at the release, it is going to rupture or kill the host cell that it was originally a part of. So this is the lytic cycle. The lysogenic cycle has the same steps as the lytic cycle. However, there's a pause in here. So we first see attachment that takes place and then penetration, just like we saw with the lytic cycle. However, after penetration, we see a new step. This step is called integration. So this is where the viral nucleic acid is going to be incorporated or integrated into the host cell's genome, into their chromosomes. All right, this is where it's going to sit dormant. It's going to sit there a while and it's not going to really do anything. Once it gets triggered, whatever the trigger is, the stress or whatever, it's going to initiate the lytic cycle. This is going to cause the nucleic acid piece to come out and start the rest of the process we saw with the lytic cycle. It's going to cause biosynthesis, where we're going to make viral parts. We're going to see maturation, where we put all the parts together, and then we do see release as well. All right, and so in this case, though, it can sit dormant for a long period of time in the lysogenic cycle. The lysogenic cycle does cause us a potential issue. Instead of in the lytic cycle, one cell being infected and then producing thousands of viruses, we see this cell goes into this dormant stage. It divides normally and makes more and more cells, and in this case, it made 4 cells. Then the stress came in, and each of these 4 cells now is producing thousands of viruses. So the infection can be exacerbated very quickly in a lysogenic cycle. Also, guys, if you'll notice here in this PowerPoint, you can click on these links in Canvas when you pull the PowerPoint up as the PDF, and you can watch these YouTube clips that really show you the lytic cycle and the lysogenic cycle in more detail. Now, some viruses are also a little more complex in the fact that they are what we call retroviruses. Retroviruses are the ones who have the RNA as their nucleic acid. So they have attachment, they have penetration, but then they also have to go through the process of going from RNA to DNA. To do this, they have to use a special enzyme called reverse transcriptase. This allows them to go backwards. Okay, we normally see transcription where we go DNA to RNA, but in this case, we're going backwards. After that, we see it enters into the lysogenic cycle for integration. Okay, it'll then stay there until it's stress, some sort of trigger happens, then biosynthesis, maturation, and release will occur. Some other things that happen here, and in this case, it's showing you like the HIV virus. When release happens here, it buds off, and it takes some of your plasma membrane with it. When it does that, it now has the envelope, and it can hide from your immune system because it looks like you. It has an envelope from your cells. This makes it harder for your immune system to overcome it. And it also does not kill the host cell to begin with. Okay, so this is looking at the replication of a retrovirus. Again, if you'll notice in this PowerPoint and Canvas, you can also watch this YouTube clip that really focuses in on how the retrovirus replication works. This chart's found in your textbook. It shows you the stages and what happens in each stage. It shows you the difference between the bacteriophage and the animal virus. Now guys, viruses a lot of times are linked to some types of cancers. This is due to the process of transduction. It's the transfer of DNA from one cell to another by the use of a bacteriophage. This may result in cellular transformations. It causes your own DNA to start to change, okay, to have a transformation to it. This can convert some special pieces of our DNA that we actually have that are called proto-oncogenes into oncogenes, meaning that they are precursor cancer genes and then they get turned into the cancer genes. Another mutation that can be a problem is if the mutation is in our tumor suppressor genes. This causes these genes to be turned off, and so tumors are not suppressed anymore and your cells will start dividing uncontrollably. Some DNA oncogenic viruses are things like HPV, Epstein-Barr or EB virus, and hepatitis B. Some RNA oncogenic, these are viruses that can actually trigger T-cell leukemias and lymphoma. All right, so when we're looking at this is in your textbook and it shows you kind of how a viral infection works. Most of the time viral infections are very acute, meaning that they are short term, they peak and then they eventually will die out and that's it. And you see that here in the red. Okay, so infection takes place, we see the acute infection happens, it increases on the chart and then it starts to decrease. However, there are some viruses that actually are more persistent. These are called persistent infections, where the first infection takes place and then it starts to go down, but then over the months and years we start to see that it still will start to increase and cause a problem. These are things like HPV, HIV, and HBV. On the other hand, other infections are considered latent infections. So once you have them, they then sit latent or dormant in your body. So then stressors or something can trigger an infection to come up later. Infections, some examples are things like cold sores, genital herpes, and shingles. A viron, when we look here, can be found with animals, plants, or bacteria. This is a complete virus, meaning that they contain a protein capsid with an internucleic core inside of it, whether that's DNA or RNA. We also then have viroids. Viroids are only seen in plants, and these are infectious RNA particles. They don't contain the protein capsid, they're just the RNA strand, and so because of this, they're not considered a complete virus or what we call a viron. So this is also known as a puron or a prion. These are found in animals. These are the proteinaceous infectious particles. These are where proteins are misshapen and they're not doing their job, and they actually cause other proteins to unfold and not be utilized and used anymore. It's almost like doing a peer pressure type of thing of saying, hey, I'm not doing my job anymore, you can unfold too, and that's what they do. Some examples of these are what we call scrappy. This is found with sheep, and then also mad cow disease, which is found in cattle or cows. One thing about these is they are not heat sensitive, meaning that when we cook our meat, even if you cook it well done, these guys can survive that process. So this is one reason why when you have sheep or cattle that have been infected with this, we cannot slaughter them in order to eat, and we don't need to then feed them to other organisms or animals either, because then it will cause that same problem. What happens is it starts to affect the proteins in the brain and causes them to misfold. It then causes the brain to start to deteriorate and look like a sponge. This can also be seen in humans when we look at Kruar and Creutzfeldt-Jacobs disease. When we look here at Kruar, a lot of times is due to cannibalism, and this is where individuals, and yes, this does still happen. We see that in some cultures, they believe in order for their loved ones to pass on to the next life, whatever it might be, they need to eat certain parts of that individual, and this normally includes the heart and the brain. And again, this could be passed on through that process. The Creutzfeldt-Jacobs disease, or CJD, can also be a genetic issue that can be present, where the proteins start to unfold due to a genetic component, which then can progress into this. All right, so if you look here, guys, viral diseases and their agents, you need to study these. There is a study sheet that really focuses on the viruses. You will need to know these diseases when you get to your final exam. So this concludes chapter 13.